Poster is about the morphologies we use here at the Blue Brain Project, and the morphologies are a crucial component of the simulation process at Blue Brain. Not only do the morphologies determine the connectivity within the cortex, but each individual morphology also affects the electrophysiology that's simulated inside that neuron. And so this poster is an overview of our morphological process, how we go from raw experimental neurons that have been filled with a dye, how they're reconstructed digitally inside a computer, and how we then repair those morphologies and prepare them for use in the simulation. Over the past couple decades, Henry Markram and his lab have collected an enormous amount of morphological data. So every time they do a patch clamp experiment, they fill the morphology with dye and then reconstruct it. And so we've got almost 1,000 reconstructed neuronal morphologies from the cortex that we can use in our simulations. And so that's fantastic. It's an enormous data set. The morphologies come from brain slices that are 300 microns thick, so anything outside of that slice region is missing from the morphologies. So we have a statistical algorithm that replaces the symmetry of the neurons by sort of regrowing missing parts. We also have a process for cloning the morphologies. We know that morphological diversity is vital for getting robust invariant connectivity patterns in our simulations, and there's evidence that that's also the case in the nervous system. So in order to get more morphological diversity from the set of actual reconstructions we have, we do a process we call cloning, where basically we statistically jitter each morphology. So each branch length is allowed to change by a random amount, and the branch angles as they come out of the neuron are rotated a random amount. And that allows us to take the 600 or so reconstructions we have that are suitable for simulation and put those into an infinite number of clones that each have a unique space filling. And that results in better connectivity pattern. What we also do is classify our morphologies into different morphological types, or M types. Right now we divide them into 55 different M types across the 6 layers of the cortex. Currently it's basically an expert process, so we have neuroanatomists who've been working with these morphologies for decades, and they go through them and based on the statistics, the morphometrics they measure from these cells, they classify them into different cell types. Currently we have our morphological experts look at the morphologies, and they say, based on these numbers and the way it's filling space and the electrical behavior, this is a morphology of this type. But we'd also like to have a completely objective classification routine that runs on the data automatically and says, well, it's 75% likely that it's this cell type. So one of our big goals over the next year is to develop computational neuroanatomy tools, which are useful not just for us but for the wider neuroscience community. So we're developing a morphological analysis toolkit in Python, essentially, which we hope to open source soon. And that allows us to import a population of morphologies and analyze them in every morphometric manner that people have talked about in the past. So basically a complete morphometric analysis on these that's easy to run and quick and useful.